Hello, welcome to the Omni-Biotic educational webinar, The Impact of the Gut Microbiome on Immune Development and Competency. I am one of your speakers, Dr. Michelle Maddox.
And I am your other speaker, Dr. Andrea Gurman.
In the interest of the ability to focus on the slide deck, we are going to stop the video of ourselves now. So the goals for our conversation are to do a quick terminology review. And from there, we will discuss the tandem development of the human microbiome and the immune system. So focusing on this window of opportunity. We will cover microbe mechanisms in the immune function, and then go into microbiome, probiotics, and potential for disease state modulation for those specific diseases listed here. And then finally, we will focus on clinical applications for the microbiome and the immune system interaction. The gut microbiome, immune development, and competence, specifically GALT, or that gut-associated lymphoid tissue. 70% of human immune system resides in the gut. The main components of this are the lamina propria, the mesenteric lymph nodes, the intraepithelial lymph nodes, and the lymphoid follicles, which include Peyer's patches. Let's review a couple of quick definitions before we get too deep. So microbiota, this is the organisms. Metagenome, those are the genes and the genomes of the population. So this is the bug and the host. And the genome is just the sum total of the organism's DNA. The microbiome equals the gene, genomes, and the products of the population. Again, this is the bug and the host. And so this is where we're looking at the whole soup, not just the individual ingredients. And to me, this part is really exciting because this is looking beyond the bugs into what they're actually doing. This is where we start getting into things like metabolomics. So we're going to cover and speak a lot about this word strain. And so for just a moment, I'm going to do a brief review. Some of you will be very bored and others of you will appreciate this. I apologize for those of you who are bored. Let's go back to our basic biology. Most of us learned some version of this picture here, right? Kings play chess on fine-grained sand. Kingdom, phylum, class, order, family, genus, species. You will notice that within this taxonomy, there's no strain listed. So what is a strain? A strain is isolated from a species and is isolated based on genotypic and or phenotypic characteristics. Specific strains are then tested for targeted outcomes. And so what do we mean by targeted outcomes? And you can see the example here. So L. casei, W56, has been shown to have a positive impact on depressive mood. However, W72 has been shown to have a beneficial impact on the immune system and no impact on depression. This particular slide is just a very quick review of the innate versus the adaptive immunity. So innate, if you remember, this is something that we're born with, whereas adaptive, this is acquired based on our life exposures. The adaptive immune system is both specific and, of course, adaptive. I always put a visual up because I am a very visual learner. And so here is a quick reminder of cells in the innate and the adaptive immune system for those who are visual like me. I love the quote, half of the immune system is designed to turn the other half off. It's a dance between reactivity and inflammation to stop the assault on the body, but then it needs to go away. If it doesn't go away in a timely fashion, we're in trouble. If it over responds to the stimulus, we're in trouble. The dance is having the perfect amount of response for the perfect length of time. And now I'm going to turn it over to Dr. Gurman.
Thank you, Michelle. We're going to switch focus now to spend a little bit of time to talk about the in-tandem development of the human intestinal microbiota and the immune system maturation, specifically focusing in on what has come to be referred to as the window of opportunity. And the window of opportunity is essentially the first 1,000 days after conception. It includes pregnancy and the first 2 years of life, and is a critical time for the development and health of the future adult. Both physiologic and immune development of the infant and establishment of their microbiota occur again in parallel throughout this short space of time. Next slide, please. Now, there is an expanding understanding of the role that a mom's immune system has on her baby's developing immune system. There are several key maternal influences, and they include things like the maternal tolerogenic immune environment that skews mom's immune system from a balance of Th1 and Th2 activity more towards Th2 activity in order to tolerate the presence of the developing fetus. In addition, the maternal system produces a variety of tolerogenic mediators like antibodies and growth factors, which pass through the placenta and impact fetal immune development. As you might imagine, the neonatal immune system responds accordingly. So in early postnatal life, the neonatal immune system gradually increases Th1 activity to restore the balance of helper T cells. Without the shift, Th2 persistence can go on to be associated with atopic disease development later in life, including asthma. In addition to this reequilibration of Th activity, neonatal regulatory T cells suppress the activation and development of naive T cells towards Th types, thereby maintaining both mucosal homeostasis during pregnancy and infancy. The key here is that this dampened pro-inflammatory cytokine response allows for the establishment of the infant's microbiota. Next slide, please. Now, as we just alluded to, the capacity to accept the developing neonatal microbiota can be explained by the relative immaturity of the neonatal immune system at birth, as well as the overall tolerogenic environment that defines early mammalian life. Hallmarks of this include, again, an overall decrease in inflammatory cytokine production and a skewing of T and B cell development toward regulatory responses. So as an example, early exposure of the host to commensals results in the repression of cells that actually induce inflammatory responses, like invariant natural killer cells. These are called iNKT cells. While these shifts allow for the microbiota establishment without the generation of overt inflammation, they also place the neonate at a higher risk for infection during this period of time. MAMPs are microbial-associated molecular patterns. They are conserved bacterial molecular motifs that are recognized by host pattern recognition receptors. In the neonate, there's this unique integration of signaling which promotes healthy microbial colonization that may be a result of the actions of epithelial-derived enzymes. Neonatal toll-like receptors and ligands respond to microbial ligands, in turn decreasing production of inflammatory mediators like oxygen-free radicals and increasing production of regulatory cytokines like IL-10. Finally, factors in breast milk like live microbes, IgA, cytokines, and immune cells are thought to be involved in creating a tolerogenic response to the developing microbiota. What's interesting is that during weaning, there's this transient, robust increase in inflammatory cytokines like interferon gamma and TNF alpha by neonatal T cells, which interestingly can then be mitigated by Treg cells that are induced by the developing microbiota. The bottom line is that disruption of this process creates an increased risk for inflammatory disease in adulthood. Next slide, please. So let's just turn our attention now to the development of the neonatal microbiome specifically. Without a doubt, an infant's route of delivery remains one of the most critical factors impacting the development of the microbiota. So the microbiota of the healthy newborn closely matches the maternal stool, vaginal, or skin microbiota, depending on whether the infant is delivered either vaginally or via C-section. The first colonizers are usually facultative anaerobes like staph species and coli, and this is followed by an accumulation of obligate anaerobes like bifidobacteria, Bacteroides, and Clostridial species within the first 6 months of life. The diversity of the neonatal microbiota is really narrow in the early infancy, and it's dominated by species which are involved in human milk oligosaccharide metabolism. These are species like Bifidobacter infantis in the breastfed infant. Now, it's been estimated that somewhere between 25 and 30% of the infant bacterial microbiota originates from breast milk, and cessation of breastfeeding, not the introduction of solid food, impacts composition as well. So bifidobacteria, staphylococci, and streptococci significantly decrease upon cessation of breastfeeding, whereas bacteria within the Lacnospiraceae family, like Lactobacterium, Roseburia, and Blaudia, increase. Members of the Lacnospiraceae family have been associated with decreased risk of atopy later in life. The key here is that succession of the early life microbiota plays important roles in the growth and maturation of the endocrine, mucosal, immune, and central nervous systems. Next slide, please. Now, as we've been discussing, the window of susceptibility and opportunity represents the period around birth when promoters of microbial homeostasis may have the largest effect on correcting microbial dysbioses. These early life disturbances are associated with colonization differences in the neonatal microbiota in the early weeks to months of life. And they've been associated with immune dysfunction and immune-mediated diseases, both in the older child and adult. Now, many of you are going to be familiar with these disruptive factors, and they include things like intrapartum antibiotics, neonatal antibiotics, C-section delivery, and formula feeding. On the other hand, neonatal probiotics, vaginal delivery, breastfeeding have all strong associations with healthy colonization and a decreased risk for immune-mediated disease development. There are other therapeutic interventions such as maternal probiotics and vaginal swabbing with probiotics, which appear to have promise as future standard of care agents. Next slide, please. This graphic is taken from a paper that sought to demonstrate the critical nature of microbial colonization in shaping the immune system, leveraging a germ-free mouse model. Specifically, the researchers looked at invariant NKT cells colonizing the lungs of germ-free mice under several different scenarios, and then monitored the animals for lung health outcomes. As a reminder, these iNKT cells are innate-like lymphocytes that secrete Th1 or Th2 cytokines when antigens bind to T cell receptors. Activated iNKT cells can regulate the adaptive immune response by recruiting, activating, or modulating natural killer cells, dendritic cells, and BNT cells. In mice, iNKT cell colonization is regulated by the microbiota in early life and influences susceptibility to asthma later in life in the animal. So in this picture on line 1, this is showing a cohort of germ-free mice with iNKT colonization in the lungs who are not exposed to any intervention, and these animals went on to develop susceptibility to asthma. In line 2, there's a cohort of germ-free mice who have been exposed to conventionalization. This is the little cluster of green microbes that you can see on screen in line 2. Conventionalization is essentially colonization of germ-free mice with the total fecal microbial community of their siblings who were born and raised conventionally. So in this slide on line 2, these germ-free mice were exposed to conventionalization outside of the window of opportunity. And these animals actually went on to develop susceptibility to asthma. In line 3, the germ-free mice who were exposed to conventionalization were exposed during the window of opportunity. And this cohort of animals went on to demonstrate healthy lung development. In line 4, another cohort of germ-free mice were exposed to monocolonization. That's exposure to one particular species of bacteria, in this instance, Bacteroides fragilis, during the window of opportunity. Unfortunately, these animals went on to develop susceptibility to asthma, suggesting that monocolonization with a single species is not sufficient enough to prevent susceptibility to asthma. Next slide, please. So as our knowledge and understanding about the dynamic gut microbiota and immune interplay or dance expands, considerations about how these insights might be used clinically, especially preventively, also arise. One area of great clinical concentration or focus is related to the risk for atopy and allergy. Now, as you remember, atopy refers to the genetic tendency to develop allergic diseases. And these are diseases such as allergic rhinitis, asthma, and atopic dermatitis or eczema. Atopy is typically associated with heightened immune responses to common allergens, especially inhaled allergens and food allergens. And it is an exaggerated IgE-mediated immune response. In our discussion of the window of opportunity and neonatal microbiome development, we've definitely talked about maternal and neonatal probiotic supplementation as promoters of microbial homeostasis. The key here is that bacteria are powerful immune modulating factors, and probiotics may exert their immune modulating effects in a variety of ways. So as an example, it may be that they exert an effect by influencing the maturation on the intestinal barrier, or by influencing the rebalancing of Th1 and Th2 responses while suppressing Th17 cells. An example of this would be probiotics leading to the stimulation of regulatory cytokines like IL-10, which suppress Th2 responses. They may also result in a decrease in the secretion of Th2 cytokines like IL-4, 5, and 13, which tend to dominate in atopic disease. Probiotics also promote tolerogenic or may promote tolerogenic dendritic and Treg cell development. Alterations in Treg or T regulatory functions are associated with the development of allergic diseases. And some evidence indicates that the gut microbiota acquired early in life is essential for the right development of Treg and Th1 and Th2 balance. We'll be taking a closer look at probiotics as modulators in atopic and other diseases a bit later in this conversation. Michelle?
Thank you, Andrea. So now let's focus on how the microbiota influence our immune competency. First, let's look at microbe containment. We want to contain the microbes to the tube in the middle of the body. Remember, our GI is a tube that runs from the mouth to the anus going through our body. And so containing microbes is very important. Remember, our immune system is aimed at controlling our relationship with microbiota and our microbiome. And microbe containment is achieved via this mucosal firewall. It's made up of the mucus layer, which is the first line of defense in the GI. This is produced by goblet cells. It's a physical barrier to prevent the host microbiota contact and translocation. Then we have epithelial cells. These produce antimicrobial peptides, otherwise known as AMPs. And these are things like antidefensins and Reg 3 gamma. This is often referred to as the demilitarized zone. IgA, specific for commensals, is produced with the help of dendritic cells. And remember, these are our antigen presenting and phagocytic cells. These interact with the B and the T cells in our Peyer's patches. This produces commensal-derived antigens based on microbiota sampling. Literally, the It goes out into the lumen and samples the microbes and comes back and presents them and says, friend or foe. Next, we have the activated T cells. These reside in tissues with increased commensal colonization, so things like our gut and our skin. Th17, which are pro-inflammatory cytokines focused on that defensive response, these cells produce interleukin 17. Interleukin 17s are neutrophils that are responsible for activation and mobilization. And then interleukin 12, which are responsible for tissue response during inflammation via proliferation and anti-apoptotic pathways. All of these support homeostasis between the commensals and the host. Again, this is this very key point and very good picture of that dance between our immune system response and the microbes. Next, we have a direct pathogen response. So remember, a pathogen is something that is not supposed to be there, or it's there in an amount it's not supposed to be there. So we have in our GI, we have commensals, and they're part of our community, right? They're supposed to be there. We live together in harmony. But these pathogens or potential pathogens, sometimes they're things that are supposed to be there, but they have to be kept in check. And if they overgrow, now they are a pathogen. There are, of course, also your frank pathogens, which are harmful at any level. And so one of the ways that we have these direct pathogen responses is via colonization resistance. And so we have competition for resources. So competition for nutrition, our food and our vitamins. There are specific bacteria that help us make some of our vitamins. And there are also specific bacteria that are dependent upon vitamins. One way to control the commensals is to control what vitamins they get. Most of us don't think very much about a B complex being part of the ways that we support our microbiome, but it is. Next, we have production of bacteriocins. These are short, toxic peptides produced by specific bacteria to inhibit colonization and growth of other species. And we all are also aware that bile acids have antimicrobial properties as well. The conjugated primary bile acids deconjugate to secondary bile acids in the colon, and these can be highly toxic to certain bacteria, things like Staph aureus and C. diff. We have our mucus layers, which we already discussed, and this is that healthy barrier, and it's that first line of defense in the GI. And then we also have short chain fatty acids. These are produced by bacteria fermenting indigestible carbohydrates. They create a hostile environment by lowering the pH. Interestingly, they also have the ability to lower the virulence gene expression in certain microbes directly. And lastly, let's look at these regulatory responses. So again, the GI starts in the mouth and it ends in the anus. And this picture introduces the oral microbiome in that middle box. The oral microbiome is highly, highly important. However, it's a very long and drawn out subject, so it's going to be saved for another day. So for tolerance in our GI, the FOXP3 Treg cells, so these suppressed cells like the NF-kappa beta, they maintain peripheral and mucosal homeostasis by sensing microbial products and metabolites. Disruption of this homeostasis results in a loss of the oral tolerance and development of abnormal immune responses in the gut. The dendritic cells, which are those antigen-presenting cells, in the gut, they produce factors like vitamin A metabolite retinoic acid for induction of these Treg cells. Short-chain fatty acids, especially butyrate, regulate the size and the function of the regulatory T cell network. So another way to say this is that short-chain fatty acids, which we get by eating fiber, regulate the cells that act to suppress the immune response. And these Tregs are critical in preventing autoimmunity. So let's look again at innate and adaptive immune functions as they relate specifically to our gut microbiome. So secretory IgA communicates the luminal contents to the lamina propria by sampling commensals and presenting them to the tolerogenic dendritic cells. So again, it goes out into the lumen, samples, and presents it back. The IgA in plasma can also express TNF alpha and inducible nitric oxide synthase. It's part of how the IgA system works to compartmentalize the microbial responses. And essentially it's walling it off to prevent a systemic response to the microbiota. Toll-like receptor, or TLR5, it's the local immunity by attracting neutrophils and macrophages, et cetera. And it also secretes cytokines. This is expressed in the basolateral surface of intestinal epithelial cells. It's the surface receptor for flagellin. So that's that structural component of bacterial flagella. These are referred to as segmented filamentous bacteria, or SFBs, and they're considered a keystone species in the gut because they interact closely with the mucosal tissue via tight adhesion to Peyer's patches and epithelial cells. This allows a heightened immunity and protective response to pathogens. And then finally, inflammasomes, which are, again, part of the innate immune system, and these are responsible for inflammatory responses. They continue the microbiota host dialogue by sensing PAMPs and DAMPs. So PAMPs are pathogen-associated molecular patterns, and DAMPs are damage-associated molecular patterns. So the microbiome and adaptive immunity. Most of this slide is a review of things we've already discussed in this section. I really like this visual. I think it's very helpful in cementing some of these concepts and the way that our immune system interacts with our microbiota. So many bacterial species can induce and regulate regulatory cells like CD4, FOXP3, interleukin 10, et cetera, in our intestines. And the microbial metabolites like short-chain fatty acids have been shown to induce generation of mucosal Tregs. The lack of a competent and adaptive immune response with corresponding changes in the microbiome can result in a negative impact on an organism's health. So in other words, these immune triggers, the responses to friend, foe, up and down regulation of recognition of self, up and down regulation of inflammation, if they do not perform the dance perfectly, we can get into trouble. If the microbiome is unhealthy, there is no way for us to perform that dance well. Now, all of these actions that we are discussing are strain specific. And that means, again, that just because we know that Lactococcus lactis is present, we don't know what strain is present, so we do not know what action it's going to have in our body. Andrea?
Thanks, Michelle. We are going to now focus more on clinical aspects of some of the material that we have been discussing in the sections above, starting with the consideration of probiotics as modulators. So remember again that there is this in tandem development of and ongoing lifelong dance between the gut microbiome and the immune system. And it certainly raises the question about how this didactic information might be translated into the clinical care of patients. As we learn more about the composition and function of the human microbiota, especially about strain-specific microbiota impacts on immune function, the possibility of using these particular insights in support of health and therapeutics in the clinical arena naturally arises. So we're going to be considering this again in a bit more on the next slides. Next slide, please. In the past 2 decades, we've learned a vast amount about the impact of the microbial flora on human health and disease. So as an example, I'm thinking here about the NIH's Human Microbiome Project, which extensively leveraged culture-independent methods for deeper levels of exploration into the human microbiome. Techniques like metagenomics, whole genome sequencing, and 16S rRNA vastly improved our understanding of host-bug interactions. In this era of precision medicine, then, scientists and clinicians are increasingly turning their attention to the promise of modulating disease expression by modulating the microbiome. And the quote on this slide is taken from a paper exploring exactly that possibility. Specifically, these authors were postulating were postulating about the possible opportunity for modulating the expression of allergic disease, specifically by modulating the microbiota. Next slide, please. In 2009, Neers et al. Undertook a study that explored the potential for modulating disease risk by therapeutic intervention during the window of opportunity. The authors considered that modification of the intestinal microbiota by administration of probiotic bacteria might be a potential approach to preventing allergic disease. And they aimed the study to specifically look at primary prevention of allergic disease in high-risk children by supporting them with either pre or postnatal supplementation of selected probiotic bacteria. This was a double-blind, randomized, placebo-controlled trial, and a mixture of probiotic bacteria was prenatally administered to moms of high-risk children. And the definition of high risk here was having a positive family history of allergic disease. So again, the mixture was prenatally administered to moms of high-risk kids and then also to their offspring for the first year of life. Of note, the bacterial strains that were used in this formulation, and they're mentioned here on the slide, Bifidobacterium bifidum, Bifidobacterium lactis, and Lactococcus lactis, were selected by in vitro experiments that assessed their ability to suppress in vitro production of lymphocytes like T cells, B cells, and natural killer cells, as well as to stimulate IL-10 production. The findings included a finding of significantly lower parental reported eczema during the first 3 months of life in the group receiving probiotics compared to placebo, as well as a significantly lower cumulative incidence of parental reported eczema during the first 2 years of life. In addition, the study authors found significantly more frequent colonization with higher numbers of Lactococcus lactis in the intervention group, and also a decreased in vitro production of IL-5, which is a pro-inflammatory cytokine at 3 months of age in the probiotic group compared to the placebo group. Next slide, please. Earlier in the presentation, we discussed the impact of dietary factors and microbial composition and function as the two primary factors that influence immune tolerance in the GI tract, and the correlation between factors that disrupt the microbiota during childhood and the development of immune and metabolic conditions later in life, including food allergy. So factors responsible for dysbiosis that have been associated with the occurrence of food allergy are depicted in the upper part of this image. And those include, again, cesarean delivery, lack of breastfeeding, and antibiotic use. Given the preponderance of C-sections in the US and increasing rates of C-section worldwide, as well as the robust use of antibiotics, not only in medical care, but in industrialized food production, it's really not surprising that over the past several decades, there have been changing patterns in the epidemiology of food allergy noted. Specifically, these patterns include increased prevalence, increased severity of clinical manifestation, and persistence of food allergy into later life. In the normal physiologic environment, macromolecules from the diet induce the bulk of T regulatory development, and this is critical for suppressing the default immune response to dietary antigens. Current evidence suggests that microbial metabolites like short-chain fatty acids are critical for the development of immune tolerance to food antigens, and in the colon specifically, Clostridial species are prominent sources of their production. Short-chain fatty acids have been implicated in the regulation of both the proportions and functional capabilities of colonic Tregs. And as Michelle mentioned previously, short-chain fatty acids play a critical role in mucosal integrity, in local and systemic metabolic function, and also in stimulation of regulatory immune responses. In general, the microbiota of kids with food allergy tends to have lower diversity, lower Bacteroidetes presence, higher Firmicutes presence compared to their healthy aged peers. Next slide, please. So we know that the gut microbiome has a critical impact not only on inducing immune responses at the local level, but also at distant mucosal sites, including the lungs. This review of randomized controlled trials from January 2010 to January 2020 was conducted to evaluate the effectiveness of probiotics in the prevention, control, and/or treatment of respiratory tract infections. In addition, the study looked at whether probiotics might also be effective against COVID-19. So there were a total of 27 clinical trials enrolling just over 9,400 patients with RTI, as well as 10 ongoing clinical trials of probiotics intervention in COVID-19 that were reviewed. So in these studies, there were 15 different probiotic strains used, and you can see the distribution of those strains on the slide graphic. Lactobacillus species were studied the most frequently, followed by bifidobacteria species, and L. paracasei and L. rhamnosus were the most commonly used probiotic strains. The review found that probiotics could significantly increase plasma levels of a variety of cytokines, like IL-1 beta, IL-6, IL-8, 12, 5, IL-10, and TNF-alpha, and also increased influenza vaccine effectiveness as well as patient quality of life during respiratory tract infections. Probiotics were also found to reduce viral titers and also found to reduce the incidence and duration of respiratory infections. So while the exact mechanisms of action of probiotics in RTI are not yet known, the authors underscored that the activity seemed to be specific to particular species and strains of probiotics. They underscored that therapeutic strategies such as the optimal dose, duration of specific probiotic species, etc., are yet to be agreed upon. And finally, the systemic review of broad clinical-based studies per the authors, given multi-center activities from a multitude of different countries, is thought to be able to progress our understanding for determining the potential use of probiotics and/or prebiotics in the context of SARS-CoV-2. So a number of those studies are still ongoing, and we should anticipate some of their findings within the year. Next slide, please. So let's turn our attention now to antibiotic-associated diarrhea. As you all know, diarrhea is a common adverse effect of systemic antibiotic treatment, especially with use of antibiotics-directed anaerobes like cephalosporins and clindamycin. And it's estimated to occur in anywhere from 5 to 39% of patients, and can manifest anywhere from the very beginning of antibiotic treatment to up to 2 months after the completion of treatment. This was a 2017 systematic review and meta-analysis, and it sought to assess the benefits and harms of probiotics used for the prevention of AAD in an outpatient, and that's the key here, an outpatient setting. The PubMed database search yielded a total of 17 RCTs with just over 3,600 participants, and a meta-analysis was conducted for the primary outcome, which was looking at the incidence of AAD. The results found that antibiotic-associated diarrhea was present in 8% of the probiotic group compared to 17.7% of the control group, which was a statistically significant difference. In addition, there were similar species-specific impact with the use of probiotic strains, L. rhamnosus GG and S. boulardii. There was also a meta-analysis done on 10 trials which reported adverse effects, and they demonstrated no statistically significant differences in the incidence of adverse events between the intervention group and the control group. And these adverse events were essentially minor events, minor in nature, and they included reports of symptoms such as having a metallic taste in the mouth, nausea, decreased appetite, belly discomfort, and headache. As a result, the authors concluded that probiotic use may be beneficial in the prevention of outpatient AAD and is likely safe. Next slide, please. Now, this study is looking at individuals who are residents of nursing homes. And we know that AAD is also a significant issue for anyone living in a long-term care facility, where it can lead to really severe complications like dehydration and malnutrition, as well as hospitalization, and increased risk for developing C. diff infection. All of these things with significant negative impact, not only on patients, but also on healthcare staff and healthcare delivery costs. So this pragmatic participatory study took place from September of 2017 through February of 2019 in 3 Dutch nursing homes. And it sought to evaluate 2 primary outcomes. One, whether changes in the occurrence of AAD by administering a multi-species probiotic to nursing home residents would be beneficial. And also to look too at facilitators or barriers to implementing a process of probiotic supplementation in daily nursing home practice. So 93 residents provided data on a total of 167 episodes of antibiotic use in patients who received either amoxicillin clavulanic acid or Cipro. And of these 167 episodes, 83 episodes occurred without the use of probiotics, whereas 84 episodes occurred with the use of probiotics. The probiotics were administered twice daily, at least 2 to 3 hours before or after the administration of the antibiotic, in order to prevent inhibition of the probiotic bacterial species by the antibiotic. The probiotics were given to the residents at the start of their antibiotic treatment up until 1 week after finishing their treatment. And the authors of the study found a statistically significant lower number of AAD episodes when probiotics were given compared to when no probiotics were given. They also noted that perceived benefits of probiotics and the prescription, not considering it a supplement, but rather actually prescribing it, which put it into the formulary by the medical staff, supported implementation of the probiotic protocol. And aversion to the intake method by residents, which included the probiotic, powder probiotic being diluted in water, as well as some concern over individual decision-making as to which resident would benefit the most, were the greatest barriers to implementation. No adverse effects to the probiotics were noted in the study. Next slide, please. This is a graphic representation of the findings of the study that I just reviewed. And as you can see, in the group of residents in the highlighted red box, in the group of residents who received antibiotics and probiotics, the difference between those who did not develop AAD, 67 patients or 80%, was statistically more of an improvement than those who did develop AAD. 17 patients or 20%. Next slide, please. Now, inflammatory bowel diseases are a heterogeneous, complex immune disorder of the GI tract that present with 2 major clinical phenotypes: ulcerative colitis and Crohn's disease. And you'll recall that UC primarily involves confluent inflammation of the colonic mucosa, whereas Crohn's disease is often transmural, patchy, and can involve any part of the GI tract from the mouth to the anus. So IBDs are believed to develop via convergence of environmental, microbial, immunologic, and genetic factors, and escalation in their incidence and prevalence, especially in industrialized societies within the last 100 years, suggests impact of the rapid changes in lifestyle, diet, and environment that we've all experienced. These, of course, impact the composition and function of the gut microbiome. So while alterations in the gut microbiota have been associated with development and progression of IBD, it's still unclear which populations of microbes are involved or how they might contribute to IBD. And also, while it's unlikely that a single microbial community would account for all disease, there are most definitely shared microbial patterns that have been noted in IBD, specifically decreased relative abundance of Firmicutes and increased relative abundance of proteobacteria. Most studies to date have focused on bacterial composition and less on role of other members of the microbiome like fungi, bacteriophages, archaea, or the virome. But both experimental and clinical trials have suggested that alterations in the microbiome have a role in the pathogenesis of IBD, with an increased abundance of Candida albicans and a decreased abundance in Saccharomyces cerevisiae being noted in IBD patients. The bottom line here is that loss of biodiversity seen in IBD results in hyperpermeability, inflammation, and increased immune responses. Next slide, please. This is another study looking at the potential role of probiotics in IBD. Specifically, this meta-analysis was undertaken to ascertain whether probiotics might be used as a potential therapeutic agent in the treatment of IBD. And the aim of the authors was to assess the efficacy of the probiotics in these diseases in both the active and quiescent states. Ultimately, 17 studies with a total of just over 1,500 patients were used. And the probiotics studied belonged to different families and were administered with a multitude of different doses. Interestingly, a total of 36 different possible agents were used in the studies. And the most frequently used included lactobacillus, the highest at 63%. Strains from the bifidobacterium family used next highest in about 58%. And about 20% of studies showing use of something called the D-Simone formulation compound, which is a substance formerly known as, or a probiotic formerly known as VSL number 3. The mean duration of the treatments was just under 5 months at 4.6 months, and the longest being 12 months, the shortest being just over 2 weeks. Interestingly, the data showed a significant remission difference in patients with active ulcerative colitis who used probiotics. Unfortunately, there was no significant difference found in the use of probiotics for the prevention of UC or for the remission of Crohn's disease. And while the review found a potential role for the complementary use of probiotics in patients with UC, the authors underscored, again, as many of these studies do, the need for homogeneous studies related to multi-strain selection and dose in order to more fully assess the effectiveness of use of probiotics in patients with IBD, especially in relation to Crohn's disease. Next slide, please. And in our last slide on this section, and as we noted in the findings of the previous slide, multi-strain formulas seem to have a higher degree of beneficial promise in support of patients with IBD. And to that end, this small open label pilot study looked at the self-reported effectiveness of a multi-strain probiotic formulation in a population of 60 patients suffering from Crohn's disease or UC. And the strains are listed here on the slide, and you can see them there. Patients received 8 weeks of a 9-strain targeted probiotic intervention. And these strains, again, were included because of evaluation for their ability to inhibit various pathogens which are associated with IBD, like C. difficile and E. coli. They had been demonstrated to improve barrier function and also were shown to induce the production of anti-inflammatory cytokines. After 2 months of use, nearly 80%, so 77% of UC patients reported reduced symptoms. and in descending order of symptom relief. Abdominal pain was most frequently relieved, followed by constipation, cramps, diarrhea, and bloating. And 59% of Crohn's patients also reported reduced symptoms, with constipation, relief of constipation being the main symptom reduction, with similar relief of abdominal pain, cramps, diarrhea, and bloating. Turning it back over to you, Michelle.
Thank you, Andrea. So by now we can see how incredibly important a healthy gut microbiome is and how it interacts and influences our immune system. And we recognize that most people probably need a probiotic to help with this. But how do we pick one? And are they all the same? Is there something that we should specifically be looking for? And what's going to prompt us to pick one product off of our virtual shelf over another product? So to make an informed decision, let's start with the basics. The product should have strains listed. When you flip that bottle over and you look at the contents, all of the organisms should be classified all the way down to the strain. The strains in the probiotics should have studies showing that they are safe and effective. There are a ton of studies out there on probiotics. It's really actually incredibly encouraging to me to see them. And a quick Google Scholar or PubMed will show you a specific strain of a probiotic and it's associated with the association with a very specific clinical picture that you're looking for. The entire probiotic formulation, so it should be a multi-species formulation, and the entire formulation should have studies showing that it's effective. Preferably, those studies have been done in humans, and we tend to think that if the individual probiotic strain has been studied to be effective for this one particular symptom, then when you throw all of those probiotic strains in together, they should then have the same efficacy in the same arenas. However, what we find when we study that is that isn't actually true. Some probiotics potentiate the action of each other, while some probiotics will actually decrease the action or completely suppress the action of other probiotics. So it's very important that the entire formulation has been studied as a whole, hopefully in humans and hopefully in that very specific clinical picture that you're wanting to work on. And finally, the probiotics need to survive their journey through the GI, and they need to have metabolic activity. And the company that you're buying from should have done research on this. So it isn't just about the billions of CFUs, because a lot of us who've been in this industry for a while have heard, we pack a lot of CFUs in there because we expect die-off. Well, I don't necessarily know that that's something that I'm comfortable with. How do I know how much die off? How does the company know how much die off? When do those organisms die off? And how does that impact efficacy? A good company will have studies on how their specific formulations do in the GI. They will have studies on how their specific formulations do in transit. Are they shelf stable? How do they know that? And you should be able to access those. So everybody always is like, well, how will I find that? That's a lot of research. Well, one, you should be doing your own research, right? You should be looking for confirmation of what the company is telling you on your own. And you should call a company. It's like any other supplement. Call them and ask them. If the company has done this research, they spent a lot of money on it, and they are over the moon thrilled for you to call and ask to show it. They want you to ask them for this information. So pick up the phone, shoot them an email, and ask them. If they are not happy to send you this, if they do not have it, if what they send you does not relate to their product, that should be a bit of a warning for you about using it. So we are all in the naturopathic, integrative, functional medicine arena. And we are all very, very, very well versed in everything starts in the gut, right? Well, else fails, we start with the gut. And I think that sometimes this message gets ingrained in us in a way that we sometimes forget, even though we've been taught, if it doesn't have a clear gut component, we tend to not focus on the gut. It's this weird kind of reverse type mentality. And I think part of it is we're so excited to do something different, but also we do get ourselves into this little box. And so my encouragement for everyone listening is to really think outside the box when you're running through your cases. And we know that there's a ton of impact. We know the gut-brain axis, we know the gut-liver axis, we know the gut-skin axis, and finally, other people and scientific and literature studies are starting to actually recognize and see all of these different things that we've known about forever. So let's take a little bit of time to review some studies that are using probiotics in a case that we may not think about using them. Okay, so I fully recognize that I said think outside the gut and don't just use them in gut cases. And the first example I show you is a gut case. So bear with me. I did this intentionally. I did this because this particular study is incredibly exciting to me. The findings of this study, it's a small pilot study. It's in a long-term care facility. 100% of the patients that were studied were colonized with a multi-drug resistant gram-negative bacteria. The study results were taken from stool samples and from inguinal fold swabs. A multi-strain probiotic was given, and this probiotic was given for 12 weeks. This multi-strain probiotic specifically targeted patients who had been on antibiotics, and it was specifically targeting this multi-drug resistant gram-negative bacteria, as well as this healthy repopulation of the GI. And what's exciting to me is the data. So you can see on this bottom graphic that at week 12, which is when the probiotic was stopped, 25% of patients were no longer colonized. 12 weeks later, that number was still dropping. So at week 20 through 24, so this is a full 12 weeks after the probiotic was stopped, we see that 100% colonization dropped to 42% of patients. It did come back up by week 36, but it still only came back up to 67% of patients. Literally nothing else was done. They got probiotics for 12 weeks. This is incredibly exciting to me because it speaks to colonization. It speaks to probiotics have a greater action in the GI than just putting bugs in the gut. It's about the metabolomics. And what would have happened if these patients had actually continued with some form of a high quality multi-strain probiotic that was targeted at maintaining a good healthy microbiome? What would the data show us then? To me, this is exciting, and this really gets me excited about this as a bottom level, like the base of the pyramid of treatment. So in this next study, we are looking at patients with type 2 diabetes. They had hemoglobin A1C over 7. They all had diabetic neuropathy. They were given 2 grams of a multi-strain probiotic for 6 months. You can see on the side here what was in that multi-strain probiotic. This was a probiotic targeting liver and detoxification. Literally no other interventions were done. They were just given a probiotic for 6 months. And what we saw was a decrease in glucose and insulin. We saw decreases in inflammatory markers. We saw lipids were improved, and we saw a decrease in leptin and an increase in adiponectin. And remember, in diabetics, type 2 diabetics, what we see is this leptin resistance. So leptin is that hormone that says you're full, stop eating. And in people who are obese, which tends to be prevalent in the type 2 diabetic population, they have a leptin resistance. The brain stops listening to the signal from leptin saying, you don't need any more food. And we also have adiponectin, and adiponectin counteracts this leptin action. It increases insulin sensitivity, and it's anti-inflammatory. And what we want to see in a type 2 diabetic is that this balance is improved. So leptin goes down and adiponectin goes up. It's exactly what we saw in this study. And again, these patients were given a probiotic for 6 months. They did no dietary changes. They did no lifestyle changes. They did no movement changes, nothing. Can you imagine if this is what we saw with a probiotic as our base formulation and our base of treatment, and then we layer on even the tiniest changes in diet or movement or mindfulness, how much more of an improvement we would have seen in these particular patients? How many of you think of probiotics as a base of your treatment pyramid when you're working with a type 2 diabetic patient? Maybe more of you will. And lastly, depression. So this is focusing on the HPA axis, that gut-brain axis. This is a huge topic, and we're not going to go deep into that today. However, what we are going to look at is a particular study that I really love to look at because it used a 4-week treatment. So that's a pretty short time frame. And it was a multi-strain probiotic targeting stress and depression and anxiety. I really like this particular study because it looked at both subjective and objective data. Studies on depression, it's a little bit difficult to get objective data. We have people do, as they did in this particular study, they did the PANAS survey, but it's still a questionnaire. And so we're trying to make it objective, but at the end of the day, a human is still telling you their impression of how they feel, which is completely subjective. In this particular study, they used not just that PANAS questionnaire, but they also used the fMRIs. And what was found on those fMRIs was an increase in the functional connectivity in the salience network. And this network is responsible for attention, perception, and cognition. And they found a decrease in the functional connectivity in the default mode network. This is the network that is responsible for distraction, rumination, impaired reasoning, and impaired decision-making. In this study, they also saw significantly more positive affect, and they saw significant improvement in hopelessness and in risk aversion. And so, this is your patient that's sitting at home, ruminating, scared to go out, and they're just kind of spiraling in. They've lost hope and they're spiraling in on themselves. And I think that we're all seeing an increase in this particular individual, especially here within these last few years. And again, in this particular study, literally the only thing that was done was a 4-week treatment of a high quality multi-strain probiotic. So again, as that base of your treatment period, this pyramid, this probiotic given, and now layering in all of the other formulations and all of the other therapies, the amino acids, the dietary changes, the mindfulness, all of these little steps that can help potentiate the action of something so simple as a probiotic to help our patients get out of their house and re-engage in a positive way with their life. Thank you so much for joining us. If you have any questions or if you want any other information or any other resources, please follow one of the links down here. You can also go to the website, which is omnibioticlife.com. And thank you for joining us and for your time and attention today.
Thanks so much.